Ladies and gentlemen, in the last video session, I've told you we're going to continue playing Lego with well-known Lego bricks. So, in the last session, you've gotten to know the phosphoacyl glycerols, meaning glycerols being esterified with one or more fatty acids and also containing at least one phosphoric acid, being the acid to form an ester with glycerol. The importance of the molecule of, hello, where is my mouse pointer again? There we are, of choline, I think was stressed. I don't know whether it was stressed enough, but this is really another one of these really important biomolecules you should memorize. And then I've told you something about this inositol, which was just a hexo alcohol. Right, from those on to the sphingolipids, where once again a new molecule has to be introduced. It's sphingosine. Now, if you look at it, this predominantly is a long chain. Well, it is not an acid, it is a double alcohol. There is a primary alcohol function, there is a secondary alcohol, and then there is this amino group here at carbon number 2. Now, this, of course, should remind you of glycerol, because if you exchange this amino group by a hydroxy group and this long chain rest here by a hydrogen, we're just talking about glycerol. So this is a glycerol derivative with, and that's the important thing, a carbon-carbon bond between the third and final of the glycerol carbons and the rest. So there is no heteroatom involved here, and actually the biosynthesis of sphingosine goes a completely different way than the synthesis of glycerol does. But if we stick to the Lego concept, then we're talking about a singly unsaturated long chain double alcohol, also containing an amino group. Now, the similarity to the glycerols and the esterified glycerols will become apparent even more so in the next slides. Okay, now if we look at the alcohol functions here at carbon number 1 and carbon number 3 respectively, then you can imagine that these alcohols can easily become esterified again. No problem, this is going to happen. The acids involved here might be phosphoric acids, they might be carboxylic acids. So theoretically, all options are open. But derived from the sphingosines are the ceramides, and this admittedly is a terrible word because there is an element called cerium, in German Cer. Now, this, of course, is something you probably did not play too much with in biochemistry, but it's important to know that there is cerium. So, Cer in German. And, of course, there is the amide, which is the anion of ammonia, so the NH2 minus ion. And a ceramide neither contains this anion, nor does it contain any cerium. So just stick to the name. It's called a ceramide. Actually, what we're talking about is a sphingosine, where the amino group, the one at carbon number 2, was acylated by one or the other fatty acid. Now, please remember, these anacetyl thingies is something you already encountered. There, where we're talking about the amino equivalent of an ester. In an ester, you've got an alcohol and an acid. In the amide version, or in the anacetyl form, then you actually have an amide group, but amide not being the NH2 minus anion, but the amide as the condensation product of an amino group and an acid, just like in the amide bond in proteins. All right, so in our, when we're talking about the ceramides, this is basically just the sphingosine being acetylated at the amino function. Now, going from the sphingosines and the ceramides to the sphingomyelins, then you see we connect both the ceramide function here and we esterify the primary alcohol with phosphoric acid, which then is also connected to another amino alcohol. Here it's an old acquaintance again. Here the amino alcohol being used to form a second ester function, or a second ester bond from the phosphoric acid, one's with the sphingosine or the ceramide, and one's with the amino function, the amino alcohol. The amino alcohol in question is choline, but that's not necessarily the case. It could be another amino alcohol. Now you'll find these sphingosines, the ceramides and the sphingomyelins, both in plants and in animals, and in the human body they're extremely important with respect to the nervous system. And actually they do play a role in insulating, for instance, the nerve channels. But let's not go into too much detail, because then once again we're far more in the land and the territory of molecular biology and not in biochemistry. Now, the most important thing here is the structural similarity of both the sphingosines and the ceramides and even the sphingomyelins to the phosphoacyl glycerols you've just seen on the last slide. Which brings us to the next point, the glycolipids or glycolipids, where we have one or the other kind of a lipid connected with one or the other carbohydrate. Now, by now you might recognize this structure here. Here we're talking about, let's go back again, we're talking about ceramide. If we remember, these were the carbons 1, 2, 3, and that was the rest. Then here is carbon 1, carbon 2, carbon 3. The secondary alcohol, once again, is not being used for anything. The amino group is being amidified, so forms an amino ester, so to say, with one of the fatty acids, just like you know it from the ceramide. And the primary alcohol here, wait a tick please. I just noticed a mistake here. Actually, there was an oxygen missing. Please note, here we've got the anomeric hydroxy group of one or the other sugar, you might even recognize that one, which forms an ether bond with the primary alcohol of our, let's go back again, sphingosine or ceramide structure. So there is not a carbon-carbon bond, there is this oxygen which just was missing on this slide again. Now, if we're talking about the glycolipids, so obviously we've got our sphingosine here, or our ceramide if the amino function is not free, and then the primary alcohol forms an ether bond to one or the other sugar. In most cases, it's not only hexoses, but in very most cases, it's either glucose or the galactose, you, I hope, remember to be the C4 epimer of glucose. Now, many of these glycolipids lipids are not based on the free sphingosine with the free amino group, but actually the an acetylated form, that is the ceramides. And these glycolipids, once again, play an important role in molecular biology and cell biology because, amongst other things, they play the role of markers on the cell membranes, essentially leading to the communication of cells with one another. Now, of course, we can get to even more complex carbohydrate structures where there's more than 3 sugars involved, and one of them is sialic acid. I hope you remember those. Then we're talking about the gangliosides. And admittedly, this is actually the door to a new world, because the chemistry of the glycolipids and all the variations you'll find there plays an important role. For instance, in certain aspects of medicine, there are quite a few storage diseases with respect to the glycolipids, or the precursors or the derivatives, etc. And if you were to go into that direction, you would have to memorize quite a few of these molecules. I really don't want to go too far into that because A, you don't want to study medicine and understand what's happening. B, actually you want to be biologists and not even biochemists. So let's leave it at that. And first, just let me show you a few molecules, and what's even more important than the actual molecular structure is once again the similarity between them with respect to their interaction with one another. So for instance, if we compare a sphingosine and a sphingoglycolipid, then please once again have a look at the polarities here. When we're talking about glycerol, glycerin, this once again stems from a German slide, hence the German word unpolar instead of non-polar. So we've got the polar part of a sphingosine here. It's not N-acetylated, so we're not talking about a ceramide yet. But yeah, I think you see the structural similarity and even the similarity with respect to the polarity. And then, of course, this unsaturated long chain thingy here, which is the backbone or the structure of the sphingosine, basically brings along one of the non-polar parts already, which under normal circumstances you just got by forming an ester of one or the other alcohol groups of a glycerol with one or the other long chain fatty acid. So I think it stands to reason that this molecule can basically undergo almost the same interactions as, for instance, our acetylated glycerols, so our glycerides. If we now go to the sphingoglycolipid, then A, you see here the amino function even has been acidified, so we're talking about the ceramide, and here we've got a sugar, as you can see, in the alpha form, and obviously this is glucose once again. Look, it's the alpha glycosidic bond between our glucose and the primary hydroxy group of our sphingosine, that is our ceramide in this case. It's alpha because it's on the opposite side of the ring as opposed to the exocyclic hydroxymethylene group of carbon number 6. And once again, I think it becomes clear that this long chain side chain of our ceramide once again contributes to the non-polar part, hence it's shown in yellow, and of course the sugar molecule here, being a glucose, once again brings along the polar component, just as the free hydroxy group of carbon number 2 is, of carbon number 3 is, sorry. This was 1, 2, 3, of course. You've already seen it on the last slides. We can have a larger sugar moiety in the interaction of, on the one hand, our sphingosines and our sugar part. So let's have a look at a slightly larger molecule. Now here is our sphingosine again. It's N-acetylated, so we're talking about a ceramide. And then you see there is several sugars involved here. The standard of glucose, of galactose, our NAC-gal, you remember NAC-gluc. So here it's the carbon number 2 amino function being acetylated. Well, there is another galactose, and this is another one you already know, the sialic acid, which once again has been N-acetylated. No, you don't have to memorize this molecule. We're just talking about one example of the gangliosides, actually several in a row, because these are 3 different gangliosides with more and more sugar moieties. So if also the N-acetylated, the galactosamine and the galactose is present, we're talking about GM1. If both are missing, we're talking about GM3, etc., etc. And of course, these molecules become completely unrecognizable if you look at them in their space-filling models, which on the other hand, of course, is important to do, because then once again we can see where there's more of the red oxygen and blue nitrogen. This is obviously the more polar part, and here we've got the non-polar strands of the side chains here. We don't know which acid R it is here, but you might remember that this R can be any long chain fatty acid rest, like in this case, this one here. And it's the interaction of these really large molecules with one another which actually connect the biochemistry on the one hand, where everything's happening on the molecular level, and the cell biology, where the interaction of cells or of cell fragments with one another takes place. That, I think, is, let's say, the more or less clear border between biochemistry and cell biology. And since I really don't want this course to become, well, just predominantly me giving you 300 molecules to memorize, the most important thing here is to know your sphingosine here, to know when that as N-acetylating the sphingosine leads to the ceramides, and that it's possible to connect these sphingosines and ceramides with various sugars using, once again, the by now familiar alpha or beta glycosidic bonds. Here, obviously, it's a beta glycosidic bond between this hydroxy group of the primary alcohol of our sphingosine to the anomeric carbon of our glucose, etc., etc. So this is not a molecule to memorize. This is a molecule in which you're supposed to recognize certain aspects. Excuse me for a moment. Sorry about that. All right. Let's have a look at these gangliosides. If we look at, well, obviously there is this large sugar moiety. Here is our N-acetylated sphingosine. Here is the structural similarity between the sphingosine and the ceramides on the one hand, and of course the glycerols you already know from the glycerides. And if you remember that I've told you that these gangliosides are to be found in the cell membranes, particularly in the outer part of the cell membranes, then it stands to reason where they interact with the cell membrane, because you might remember from cell biology what the cell membrane looks like, that the cell membrane, which you know is this the lipid double layer, it's got a polar outside, a polar inside, because in both sides we've got water as a solvent, and then there is the non-polar part of the hydrocarbon chains interacting with one another, forming this double layer. And of course, this non-polar part here, once again shown in yellow, can easily interact with the other non-polar part of the cell membrane. And so this polar part, because we're talking about hydroxy groups here, hydroxy groups can form hydrogen bonds, you remember that, I hope, then of course can and will interact with the polar parts present at the outside of the cell membrane. Meaning the hydrophobic part of this molecule is anchored in the cell membrane, or you could say it is part of this cell membrane. So we will return to that, of course. But first, let's have another look at glycolipids. I've already told you, if we're talking about glycolipids with more than 1,3 sugars, and one of them having to be the sialic acids, those are the gangliosides. Now here, it would be nice if you were to memorize sialic acid, but it's not vitally important. Whereas for biologists, you should definitely know what gangliosides are, not necessarily down to the absolute molecular level with the stereochemistry, etc. But you should know that there are sugars involved, and that there's quite a bit about sugars you should memorize, etc. Okay, now if we're talking about the sialic acid, the problem is sialic acid is a whole family. There is not one sialic acid molecule. There are all derivatives of the neuraminic acid. And if we look at the neuraminic acid, then this is an amino sugar with an overall C9 body. There really are carbon-carbon bonds here, not hydroxy groups. These neuraminic acid does not occur in the free form in nature. Many derivatives do, but not the free form in itself, where you've just got the hydroxy group or the free amino group or something. If the amino group is acetylated, as you can see here, we're talking about an acetyl neuraminidate. This is also a sialic acid. So the sialic acid, like I said, is a family of molecules, and particularly the N-acetylated ones are typical signal molecules or carriers of signal molecules. So just as an example, they might interact with the important neurotransmitter of calcium ions, calcium 2+. And then, of course, they can also interact with macromolecules, where once again we leave the area of the normal biochemistry and enter the field of cell biology. I think I've stressed that now often enough, and if there are any questions regarding that, please let's discuss that in the exercises. Okay, now there are two more things I would like to briefly mention before I return to the cell membrane, which will happen in another video. Because there are terms you know from everyday life, but in biochemistry they are slightly more precisely defined. When, for instance, talking about waxes. Now, we already have the differentiation between the oils and the fats, with the only difference between the fats and the oils was that the oils were liquid at room temperature, whereas the fats were solids at room temperature. And obviously, it's possible to melt a fat so it becomes an oil, but then it's not called an oil because at room temperature it would be a solid. You might notice that this is not a particularly scientific notation. And the same term goes for the same applies to the term of waxes. There's one thing important all waxes have in common, and that is we're talking about long-chain carboxylic acids. Yay. On the other hand, these carboxylic acids form esters with long-chain alcohols, leading to extremely large and extremely heavy molecules. If you look at this one, for instance, we've got a C26 carboxylic acid, so 26 carbons. It's completely saturated here. And this long-chain carboxylic acid is connected with an alcohol, a primary alcohol, containing 30 carbon atoms. So we're talking about really heavy and really long chain molecules you'll find as protective coatings. Once again, both in plants and in animals. Now, occasionally, this myrisyl cerotate, this is okay, myristic acid is something you might know or don't. It's really not one of the molecules you have to memorize. If, on the other hand, you take the cetyl palmitate, now palmitic acid is a molecule you should be familiar. And so you see the length of both the carboxylic acid and the length of the alcohol involved being esterified with the corresponding acid can vastly vary. But you can imagine that the van der Waals interactions the cetyl palmitate can undergo are exactly the same as with myrisyl cerotate, only that, of course, the longer the chain, the stronger the interactions, the more solid the waxes will be, etc. Okay, but basically, if we're talking about waxes, long-chain carboxylic acid can forming an ester with a long-chain alcohol. Which alcohols are involved would once again go far beyond the scope of Biochemistry 101, but now I think the term waxes has become clear as well. Let's talk about steroids. And actually, it should be called Let's Talk About Steroids for the First Time, because they will return again. Once again, these steroids are also considered to be lipids. They're also part of the cell membrane. I will tell you something about in the next video session. And the important thing about the steroids is they all have the same fundamental structure. This system of 3 6-membered rings and 1 5-membered rings, which are fused so that they are that 2 rings always use 2 bridging atoms in both rings. There is not more nor less than that, so it's just a connected carbon-carbon bond, both between rings A and B, B, C, and C and D. But then the various substituents attached to the various atoms numbered from 1 through 17 here, with the numbering being a little bit of a science of its own. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, so far so good. And then 11, 12, 13, 14, 15, 16, 17. So you describe a circle and then you do this other thing here. Well, actually, you as biologists should just memorize that this here, this upper part of the 5-membered D-ring, is position number 17, because that's where where part of the magic lays in the various steroids. You might know, at least by name, not by the structure, but I think you probably have heard of the word cholesterol. You might know testosterone and progesterone and estradiol, 3 of the hormones associated with sexuality. So they're even called sex steroids in English. Okay, and all of those, and the cholesterol, which you probably will first connect with food and saying this is high cholesterol food. As you can see, they all have the same basic structure. Yes, they're all steroids. Now, the synthesis of that is something we will be interested in later on. How is this 17 carbon atoms containing fundamental structure of all the steroids really being formed? But we'll come to that one later on. The important thing for you right now is to see that it is just a question of the substituents on this structure, which, for instance, shows the difference between the male testosterone and the female estradiol. You might see in the testosterone, you have a keto group here and just one double bond, which is in direct connection with the other one. So these pi electrons are delocalized to a certain extent. In estradiol, you even have a real aromatic system. But apart from that, there is just one methyl group missing, which is not present in estradiol, but it is in testosterone. And so it is in progesterone, which also plays an important role, once again, in questions regarding gravidity, for instance. All right, we'll come to that one later on. Once again, for technical reasons, I will have to cut this session short for now, but more to come.